So where we left off on Friday, we were talking about osmosis, which is the diffusion of water across a selectively permeable membrane. So we left off talking about an example. A marine algal cell is put in fresh water. What's going to happen to that cell? And we determined that the cell would absorb water from the surroundings and expand in size. So, and the reason for this is that water will diffuse passively across the membrane from a region of lower solute concentration to a region of higher solute concentration. You know, interestingly, that process, the putting a marine organism in a freshwater environment, is actually a technique that aquarists use. It's called a freshwater dip. So aquarists that have these large saltwater tanks that maybe have sharks and large fish in them, oftentimes those large fish, those sharks, they will get parasites on their bodies. And one way to get rid of parasites really easily is they take the fish out of the tank, put it in a freshwater tank for a few hours. It's not enough time for that stressful environment to cause harm to the large fish, but it's plenty of time for it to kill off the parasites. And then they put them back in the salt water and they go about their way. You can do the same thing the opposite with a freshwater fish. You can put them in a saltwater bath. It's usually a little more stressful, so you might not do it for very long. But it's a way to kill external parasites on the organism. And that happens because those parasites, which are saltwater parasites, will absorb lots of water, they'll burst, and then they fall off the fish. So that is the process of osmosis, diffusion of water across a membrane. So it's important for organisms to be able to balance their water uptake and loss because it allows them to survive. So if you are a saltwater organism, you don't want to end up in freshwater. That could be bad for you. You could explode, right? Same thing if you're a freshwater organism, you don't want to end up in saltwater. And this can be problems, like when we've had hurricanes and we've had large amounts of ocean water spill over into freshwater environments. It can be really bad for the organisms living there, as they're now in this brackish, higher salt concentration, and it can be very stressful for them. So that has happened before, especially if you have freshwater ponds really close to saltwater areas. So the cell survival depends on balancing water uptake and loss. The word that we use to measure the relative concentrations of solute on two sides of a membrane is something called tonicity. And if you look at a cell here in a solution, and here I've just drawn some sodium molecules for reference here, but let's say I have a solution of 40% sodium chloride, and I have a cell which inside of that cell is also 40% sodium chloride. This is a situation that's referred to as isotonic. The cell is isotonic to the solution. The solute concentration is the same on both sides of the membrane. So a solution or a cell are isotonic if the solute concentration is the same as the other side of the membrane. And in this case of isotonic situation, are you going to have any net movement of water across this membrane? No, they're at equilibrium, so the amount of water that leaves the cell is going to be equivalent to the amount of water that enters the cell. If instead I had a little bit less salt on the inside of this cell, so let's say instead of 40%, this was 20% sodium chloride inside the cell, and I had 40% sodium chloride outside the cell, the outside of the cell is now what's called hypertonic. So hypertonic is when the solute concentration is greater than the other side of the membrane. So this is the hypertonic side of the membrane. When you have a hypertonic side of the membrane, you're going to have the other side of the membrane have less dissolved solute, and this would be called hypotonic. Hypotonic is when the solute concentration is less than the other side of the membrane. So I have the hypotonic side inside the cell, and I have the hypertonic side is the solution, the outside of the cell. So this is said to be a cell that's put into a hypertonic solution. What's going to happen to water in this case? So is it going to flow in or is it going to flow out? So I've got more salt outside and I've got less salt inside. Anyone? Okay, and it'll actually flow out of the cell. So water will always move from the hypotonic to the hypertonic. So this would be the case that you put a cell in a saltwater bath. So this is when you eat lots of salty foods, your blood becomes hypertonic, and so your cells all will lose water to your bloodstream. And that's why you start feeling thirsty, is that your cells have lost water. So this is, you're going to see water, H2O, now flow out of the cell. Water always goes from the hypotonic to the hypertonic. If instead I had this reversed, if I had 20% outside and 40% inside, then it would flow in, yeah. So then I would have the inside of the cell would be hypertonic, and the outside of the cell would be hypotonic, right? And I'm going to have water flow into the cell. So this is when you give a patient an IV of fresh water, you are immersing their blood cells in a hypotonic solution. This is also when I said what happens to runners that drink way too much water. They dilute down their bloodstream, their cells become hypertonic to their blood, and water enters the cells and they expand, and this can make them explode and become anemic. So again, water will always move from the hypotonic side of the membrane to the hypertonic side of the membrane. And remember, that's hypotonic is where you have less dissolved solute. Hypertonic is where you have more dissolved solute. So, another way of thinking about this, which might help. So you're seeing percentage of salt here. I've got 40% inside the cell. I've got 20% outside the cell. And so this isn't really reality, but we can consider this. You've got 60% water inside and 80% outside, so you've got a higher concentration of free water outside. It'll move in. So if I had open sodium chloride channels here, this would diffuse out. The sodium would diffuse out, but because this prevents sodium from flowing out, water is going to flow in. Okay, so hypertonic, hypotonic, isotonic. People feel okay with those words? Hyper means more dissolved, hypo means less. Water goes from the hypo to the hyper. Okay, so why? So let's apply these names to actual cells. So here is animal cell, like a red blood cell. Animal cells prefer to be in isotonic solutions. So in an isotonic solution, the amount of water leaving the cell is equal to the amount of water being absorbed by the cell. So you've got the same concentration on both sides. So this is why we give patients a normal saline at 0.9% solution, because the concentration inside the cell is equivalent to 0.9% sodium chloride. So your cells are going to lose just as much fluid as they gain, and they're not going to change in shape or size. If we put a cell in a hypotonic solution, so this is giving a patient an IV of fresh water, that water is going to flow in at a greater rate than it flows out, causing the cell to expand. Yep.
I have a question. When people are drunk or intoxicated, they always say that saves the hospital to put like saline or something to calm them down. What exactly does that do to them?
Well, it just increases the amount of fluid that's in their cells. Yeah, I don't know if it'll calm them down necessarily, but it increases their ability to process things, because they tend to lose fluid. Okay. You probably noticed drunk people tend to pee a lot. It's because one of the byproducts of alcohol consumption is that it inhibits something called antidiuretic hormone, which prevents you from losing lots of water. So by inhibiting that, you start expelling a lot of water. So your body retains less water, you become dehydrated, and you actually drink more. So by putting an IV in, that keeps the volume in their blood high enough so their body's able to metabolize all the alcohol faster. So that's what happens. They become dehydrated. Drinking alcohol is not going to hydrate you. You don't drink beer to rehydrate because it actually causes your body to lose water. Yeah. So they would actually be in this state more so because their cells would be losing water due to the effect of alcohol on that hormone. So the third state is a hypertonic solution where your cell is going to lose water and shrivel and shrink. This would be if you gave a patient a saltwater bottle, saline, and so something that's higher than 0.9%, you could actually cause them to dehydrate. So even though you're giving them fluid, you'd be dehydrating them. So we want to give them an isotonic solution so that the cells don't gain water, they don't lose water, because this is where your cells will function the best in. They'll be able to do all their normal processes. Okay, so an animal cell, isotonic solution is ideal for them. This is actually different for plant cells. Plant cells prefer to be in a hypotonic solution, and the reason for this is that plant cells have something that animal cells don't have, which is a cell wall. So when plant cells expand, because they are in a hypotonic solution, so hypotonic would be this situation. They're in a hypotonic solution. Water is going to rush into the cell. It's going to go into that central vacuole, expand the central vacuole. The cell membrane is then going to be pushed against the cell wall. Plant cell is not going to burst, though. An animal cell might explode under that force, but a plant cell has a cell wall, which prevents it from exploding. And by pushing against that cell wall, it makes the cells more solid, more rigid, and allows the plant to stand upright. So a plant that looks like this, it's well hydrated, it's actually interacted with a hypotonic solution. This is why you water your plants with fresh water. You wouldn't pour saline on your plants. That would not be very good for them. And you definitely wouldn't water them with salt water. This is actually a problem with irrigation systems. So a lot of irrigation systems use groundwater, which have a lot of minerals in them. And you irrigate a farm, and what happens is the plants take up some of the water eventually evaporates, but the minerals that were in that water that you irrigated with stay behind in the soil, and you're gradually increasing the mineral content in that soil, making it more and more hypertonic. And at a certain point, your soil is going to be so hypertonic to the plants that the plants are all going to lose water. So excessive irrigation leads to plants losing water and wilting. And that is bad. So, any of you seen the movie Idiocracy? Where they water the plants with Gatorade, right? Yeah, electrolytes, because I think, electrolytes are important, right, for everything. No, that's what would happen. This is why their plants didn't grow. It's because they were adding a hypertonic solution to the plants. So your grocery store would do very poorly to spritz Gatorade on their plants. But what they do is they spritz fresh water on the plants, and it causes the plants to perk up and look crisp and delicious, and you buy your lettuce because of that. So even an isotonic solution is not going to lead to this much wilting, but it's also not going to make your plants perky. It's going to make them kind of a little bit droopy. They're not going to shrivel up and dry up, but they're not going to be fully solid. They're going to be slightly droopy, so you don't even want an isotonic solution. So plants prefer a hypotonic solution so they can use the cell wall as their skeleton. Right? That's because pretty much any other liquid is something dissolved in water for the most part. So you'd want something that's pure, that doesn't have a lot of dissolved solids in it. So you don't even want just any fresh water. You want fresh water that has a fairly low mineral content.
Could you use like the flavored water in the store and stuff?
Well, that has sugars and stuff dissolved in it usually. Yeah, so that would also not be good. So Any of that stuff is not ideal for plants. And there's different levels of stress. So plants do have a tonicity. They do have a certain amount of dissolved solute in it. And as long as you're below that, you're putting a hypotonic solution on it. So you might be able to get away with some dissolved thing. That's why you don't need deionized distilled water to water your plants with. But you also don't want water that has a lot of minerals in it. You want water that has a fairly low mineral content, which doesn't typically happen with irrigation water. Rainwater tends to be fairly low mineral content because it's water that has evaporated and now come back down to earth. So this is why rainwater doesn't cause the same problems that irrigation water does on farmlands.
Does whatever the water cycle, does that take the minerals out?
Yeah, well, when a lot of the water that comes down in rain is water that's evaporated off of either the land or off of the ocean, and when it evaporates, it leaves behind those minerals. So the normal water cycle, rain falls on the mountains, it picks up minerals as it runs downhill into the streams, and then those minerals get left behind in the ocean or the stream beds as it evaporates again. So water coming out of the sky is pretty pure. It has very low mineral content. But groundwater has very, very high. So farmers have to be really conscious of this, and that's why a lot of farmers will rotate crops, because there are certain crops that do deal really well with high mineral contents and actually will pull the minerals out of the soil. So certain legumes, a lot of farmers will rotate in certain types of plants. And so they'll have one type of plant one year, then switch it over to one that's going to pull up those minerals the next year, and then they can go back to the first crop again. So farmers that don't do that, they have fields that are all of a sudden not growing anything the next year. Yeah? Okay, so hypotonic ultimately. So it's a very interesting thing. Okay. There's one more kind of topic. Well, actually, there's two more little topics to this chapter. I just wanted to go back briefly to this slide before we go to the next concept. So simple and facilitated diffusion. Anything that is diffusion is a passive process. It does not require energy input on the part of the cell. Simple diffusion is without carrier molecules, so it's things like oxygen or carbon dioxide just flowing through the cell membrane. Facilitated diffusion is flowing through a protein, but in both cases, you're going from an area with a high concentration to an area with a low concentration. So this arrow is going in that direction. Right. What if I wanted to take this molecule and move it that way? What would I have to do? Okay, I'd have to apply energy, right? I'm moving something uphill. This is like taking water from the bottom of the dam and trying to move it back up to the top of the dam. It'll move this way without any extra energy put into it. But if I want to move it this way, I'm going to require energy. So your cells often want to move things against their concentration gradient. And this is a process called active transport. Active transport requires energy. So simple and facilitated diffusion do not require energy. They are dissipating an existing gradient. So what that part of that sentence means is that you're taking a gradient where you have a high concentration and a low concentration, and you're allowing them to flow to dissipate that gradient to make it more equal on both sides. So that's what dissipating a gradient is, when something moves from the high concentration to the low concentration, which is basically equaling out the concentrations. Active transport does the opposite. It moves things from a low concentration to a high concentration, actually creating that gradient. And that process requires energy. Okay. So here is an example of active transport. This is the sodium-potassium pump here. And you've got sodium here in purple, potassium in yellow. And potassium's really concentrated on the inside of the cell. Sodium's highly concentrated on the outside of the cell there. And as you pump sodium out and potassium in, it burns ATP. So this is going to require energy to move the sodium out and the potassium in. So this is active transport. And your cells do this a fair amount. And we're going to talk about these in the next two chapters. Look at there. Okay, so this doesn't make it completely obvious here, but look at the ratios at which the sodium-potassium pump functions. So sodium-potassium pump has actually a large role to play in your neurons firing, and you'll probably talk about this more if you take physiology, but it plays a large role in this because it sets up this concentration gradient, but also because it sets up another form of potential energy. If you look here, we've got 3 sodium molecules, 3 ions of sodium being pumped out, and when the pump allows potassium to come in, it pumps 2 potassium molecules in. So 3 sodium molecules out for every 2 potassium molecules going in. So is the charge on the membrane going to change as the sodium-potassium pump works? Yeah, so it's pumping 3 positives out for every 2 positive coming in. So it's actually going to be losing electrons from the outside. It's going to be kind of gaining protons on the outside and losing protons from the inside. So the outside of the cell is going to become positively charged and the inside negatively charged, right? So this builds up a positive charge on the outside and it's removing the positive charge from the inside, so it's going to make it slightly more negative, right? Okay, so your neurons do this. They'll set up this pump, will pump, 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 pump until you have a lot of sodium outside and a lot of potassium inside, and you've got a positive charge on the outside and a negative charge on the inside. So now your cell is getting a signal to fire. And what that does is it causes this cell to open up a sodium channel. And what's going to happen when a sodium channel opens? So what's going to happen to sodium? It's going to go into the cell or out of the cell? It's going to go in. So the sodium is going to rush into the cell. And it's going to rush into the cell partly because it's highly concentrated outside and not very concentrated inside, but also there's a negative charge here and sodium's positively charged. It's going to be pulled in. What's going to happen to this negative charge as those positive ions rush in? Yeah, this is going to become positive and this charge is going to become negative, right? Because it's losing all those positive charges. So you flipped the charge. And now if you want to stop the cell from doing this, your cells can open up a potassium channel, which causes potassium to rush out, and that will kind of neutralize the charge. Okay? And then the sodium-potassium pump will start again to build up the positive charge on the outside, the negative on the inside. If it wants to fire again, opens up sodium channels, sodium rushes in, swaps the charges, and then potassium rushes out and neutralizes. This is how your nerve cells fire and allows them to very rapidly, so they don't have to run a transport protein from one end of the cell to the other to move a signal along. They use this and it very rapidly passes a signal from one end of the cell to the other, and this way your nerves can fire, transporting messages from your brain to your fingertips and your toes almost instantaneously. So it's a very, very rapid process. Right? So that's the sodium-potassium pump. So the sodium-potassium pump sets up something called membrane potential. So when ions are moved against their concentration gradient, there's 2 types of membrane potential. So 2 types of potential energy that are built up in the cell. One is chemical potential energy. This is just the concentration gradient. So sodium's concentrated outside the cell, potassium's concentrated inside the cell. And the second one is electrical potential, where you have that positive charge on the outside of the cell and a negative charge on the inside of the cell. So this is membrane potential. And membrane potential can be used for a variety of things. Like your neurons, it can be used to rapidly pass messages, or rapidly transport a signal from one part of the cell to the other by changing the charge on both sides of the membrane. Another thing it can do is what this bacterial cell does. So in bacterial cells, they often will pump hydrogen out of the cell, decreasing the hydrogen concentration and also decreasing the charge on the inside of the cell. And then they'll use this gradient, both chemical and electrical gradient, to do work like pumping sucrose into the cell. So sucrose might be pretty concentrated inside the bacterial cell and might be not very concentrated in its environment, and it wants to bring in more food because it wants to use that to create ATP. So they'll use something called a sucrose hydrogen ion cotransporter, which uses the energy of diffusion of hydrogen to pull in sucrose. Sucrose is also attracted by the negative charge on the inside of the membrane and can pull things into the cell like that. So it can do work this way. We'll see that this same concept, a proton pump that's driven by some energetic process, is going to set up a concentration gradient that will allow your mitochondria and will also allow chloroplasts in photosynthetic organisms to generate ATP. We're going to see that in the next 2 chapters. But it's all due to pumping ions across a membrane, creating potential energy in the form of chemical potential energy and electrical potential energy due to the concentration gradients and the charge difference. So both of those are membrane potential. Okay? So we'll see this again. We'll definitely see proton pumps again. And you probably, in physiology class, would learn about sodium-potassium pumps again. Okay. Last little section of this chapter talks about how to get large molecules across the membrane. So we've looked at ions, which are just single atoms. We've looked at small molecules like water, which is only 3 atoms tied together, or even things like small, like a monosaccharide or a disaccharide, which are still fairly small molecules. All of those, if they're charged, if they're polar or they're ions, they can move through transport vesicles. If they're not polar, like oxygen gas or carbon dioxide, they can just diffuse through the cell membrane. But if things are larger, things like proteins, things like polysaccharides, things that are made up of large amounts of atoms, large biomolecules, they're not going to be able to diffuse through the membrane, even with a transport protein, because they're just too large to fit through a transport protein. So they're going to move across the membrane in bulk by vesicles. And the 2 processes for moving things in bulk across the membrane are called endocytosis and exocytosis, and they only differ in which direction it's happening. Endocytosis is when the cell brings something into itself through a vesicle, and exocytosis is when the cell releases something with a vesicle. So the cell engulfs fluid in large molecules through endocytosis to bring them into the cell, and the cell uses vesicles to transport large particles out of the cell through exocytosis.
So like with the cell when it was taking in sodium and then like pushing out, or what was it?
Potassium in, sodium out.
Is that-
No, because those are ions. So those are small. So that's a protein pump that's pumping it across. This is when you have large biomolecules, so large proteins that are made of many, many amino acids, large polysaccharides that have many, many sugars tied together. So the sodium-potassium pump, those are just single ions. So you have like an ion, which might be that, versus a protein, which might be huge. Yeah. So this is moving large stuff across rather than small. So this is like you can, I don't know. Yeah, it's small versus large. It's just
And
size.
this
Yep.
is another form of active transport?
Trying to think. This will still, yeah, I'd say this is most likely to use energy. But again, that always depends on the concentration on both sides. If there's potential energy for something to move in. Typically, we talk about active and passive transport when things are moving through channels, versus this is a pretty active process because you're engulfing something, but you may be moving things up or down their concentration gradient. But yeah, it often does take some energy, especially when you have receptor-mediated endocytosis, where you're bringing in specific things based on how they interact with proteins on the surface, so it can get really complicated. Yeah, but we usually don't talk about active or passive when it comes to this. But you're right, it could be active. It could take energy. Okay, here are some pictures of this, just to give you an idea of what this looks like. So on the left here is endocytosis. So this side is the outside of the cell. This is the inside of the cell. Here's a bunch of molecules that need to come into the cell. The cell membrane will pinch off a vesicle, and it'll be moved into the cell. This then, so let's say this was polysaccharides, a bunch of polysaccharides. This might merge with a lysosome, and the lysosome will perform hydrolysis reactions to break this down into monosaccharides, and then this vesicle might make its way to the mitochondria to further break that down and release the energy in those fuel sources. Might also be that this was a bunch of large proteins that your cells are absorbing from food that you've eaten, and this vesicle will now make its way probably to a lysosome again, and then to your ER to drop off those amino acids that have just been produced by breaking down the protein, and it can make new proteins there. So that's endocytosis, bringing something into the cell. Exocytosis is the opposite, so this might be a vesicle filled with proteins coming off the Golgi apparatus. So remember, proteins made in the ER, transported to the Golgi. Golgi apparatus then can release vesicles to the cell surface. This vesicle would merge with the cell surface and then release those molecules, allow them to leave the cell that way. So this is also how a cell increases the membrane on the surface. So if a cell wants to grow, it's got to merge a vesicle from the ER with the cell surface. And as it adds a little bit of membrane to the surface, this one instead pinches off a little bit of membrane. So your cell needs to balance these two processes so that it doesn't change shape if it doesn't want to. But take-home message, endocytosis absorbs large biomolecules, exocytosis releases them. Okay. So here's just one more question, just to make sure you were paying attention through this. So sodium-potassium pump requires energy. Why is it? Why does the sodium-potassium pump require energy? Okay, last few responses. All right. Okay, so one of the points of having this question up here is to remind you to read questions carefully as you take the test on Wednesday. And to make sure that you're not just picking answers because what I say in the answer is true, but you're picking it because it actually answers the question that I ask. So pretty much all of you chose either C or D. So which means that, to me, that you know that C is right. You know that C is a reason why it requires energy is because it moves solutes against their concentration gradient. Which you're absolutely right. It does move solutes against their concentration gradient, and that is going to require energy. So some of you, a fair amount of you, 53% of you, said D. So which tells me that you thought A and B were also reasons why it requires energy. So it uses a membrane protein and it transports ions. So do you know of any other mechanism of allowing things to cross the membrane that can transport ions and that uses a membrane protein that does not require energy. So so simple diffusion does not do either of those things. What is the one that uses a membrane protein? Facilitated diffusion, right, the other one. So facilitated diffusion uses membrane proteins, can transport ions, and does not require any energy. So although these are true about active transport, they're not a reason why it requires energy. The reason why it requires energy is C, that it moves solutes against their concentration gradient, and that's the real point. So just be careful on Wednesday. When I ask a question, don't just jump to conclusions because they're right, but because they answer the right question. So read the questions carefully. Ask me about them if you don't make sense. Okay, that's all I have. If you have questions, I've got like 4 more minutes of class, so you're welcome to stick around.